to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 87th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for around 30 years. I'm here with my co-host today, Dawn Dufield, because Dominic Marino has a prior engagement. Dawn?
Yeah, hi, Dawn Dufield. I'm the scientific officer for the company. Been here, mostly scientific officer for mass spec. Been at KCAS for about 7 years and come from a pharma background for 20 years before that. And really my area is all things mass spec, but specifically large molecule mass spec is a big area. And Dom and I, battle a lot, so it's nice to take over his spot and, run the show a little bit.
Don and I are members of the growing scientific advisory team at KCAS Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio's services. Today we're here with a guest, Rahul Baghla, Senior Manager, Global Scientific Marketing and Ecocenter of Excellence at Sciex.
Hi, everybody. My name is Rahul. Thanks, John, for the introduction. I'm Senior Manager for Global Scientific Marketing, Pharma and Biopharma, Sciex, along with Ecocenter of Excellence, which is focused on high-throughput solutions based out of Framingham. My association with Sciex is close to 12 years. Used to be a scientist before switching over to my new role, now managing a group of scientists, basically responsible for driving global collaborations in pharmaceuticals field.
We're excited to have you here listening to the 87th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcasts, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you're joining us for the first time or if you're a regular listener. Today's podcast will be a little bit different. We're going to have a discussion of some upcoming conference news, and then we're going to focus on the topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're excited to have you here, and we're looking forward to the fun episode today. To kick us off for podcast number 87, we're going to start with a brief discussion of our plans for the upcoming WRIB meeting in New Orleans before moving on to the main topic, which is a discussion of the evolution of Sciex' partnership with Pharma in drug discovery and development. Let's move on to talking about WRIB. We're now into conference season. I just got back from SOT in Orlando last week, which was a pleasant, it was nice. As bioanalytical lab, we're a little bit on the fringe of things with it being mainly toxicology focused, but still you make contact with some some different people, and it's always good to spread the contacts we have. But the big meeting upcoming for a lot of folks is actually WRIB in New Orleans from April 7th. I think we have a pretty large contingent of going from KCAS, and I know Dawn's one of the participants going from our organisation. So, Dawn, do you want to just start and say who's going and what the plan is for the KCAS team? I assume we'll have a decent sized booth and representation from a bunch of different groups, and obviously we'll be hoping to make contact with a bunch of customers.
Yeah. So we actually have a handful of people going, a lot of people from the business development team to obviously meet with our clients and be able to answer questions. We have, I think, at least three of our scientific officers or scientific advisors. So myself, Dominic, and Lindsay, I think, are going. And we'll be there. We have a couple posters from some of the people in operations on various topics, and then I think we even have a panel interview with Bioanalysis Zone, I think, on PK, PD, immunogenicity, kind of talking about all the different support lines and technologies that we're doing. So yeah, it should be really, really good week. And then we also finally follow it up with Friday. There's a GCC, the Global Consortium Council, that does all of the CROs that kind of talk and align on recommendations for FDA and regulatory authorities and such. So should be a good week.
Do we know if there's likely to be any FDA participation in WRIB this year? I mean, I know SOT there was certainly there was people from governmental organisations was way down. I think we met one guy from the
I mean, I
UK.
expect it to be. It
Yeah.
always has a very large FDA and or regulatory component from various, you know, different countries, etc. So I expect it to be there, but it might be lower than normal just due to the environment kind of going around right now. But I do fully expect them to be there.
Okay, good. So, Rahul, is there much planned from Sciex' perspective, or is it more an opportunity to meet up with customers, or how does Sciex approach this meeting?
Yeah, so from SciEx point of view, yeah, definitely we'll be there. We'll have a booth there, but this time probably our focus is more on meeting people and increasing basically our network with the people. So we have a very like a variety of expertise going to WRIB this time. We have a scientific expertise team from tech marketing is going, and at the same time we will have market development people on WRIB as well. We have global market development team as well as our North America market development team as well. So they all come from heavy scientific background. So yeah, focus is basically networking and seeing more people around this time at WRIB.
Yeah, maybe to add to that, we do have a, I guess call it a collaboration. We've been working with Sciex for many years, but recently more focused on some marketing things. And we have some of our marketing folks going to WRB, and I think it'll be the first time maybe they all meet in person as well. So I think we're going to meet up with the Sciex team there.
Which I'm sure we'll touch on in the main topic. So
Yep.
I mean, we got plenty to talk about, and I suppose at that point we've got nothing more to say about WRB apart from the fact well everyone's looking forward to it. I mean, it'll be a quiet week for me while everyone else isn't bothering me, so it'll be good from that perspective. I mean, I suppose we can move on to the main topic. So we have a special guest, Rahul Baghla from Sciex is here to join us. He's the Senior Manager, Global Scientific Marketing and Echo COE. And we want to get together just to talk because we've worked with Sciex many times over the years. And like Don mentioned, we've had an active recent collaboration with them. And so we thought it would be a good time to get together, do a podcast together and talk about the evolution of Sciex' partnership with Pharma, and I include us as a CRO in that, in drug discovery and development. So, first of all, I'd like obviously would like to introduce Rahul Baghla. Rahul, would you like to give us a sense of well your what is your you give us a sense of your background and also what's your current role at Sciex, and just give us some of your personal history and what got you to where you are today. It's always interesting to hear about our guests, what they've done prior to their current role and what their current role is. So if you want to tell us all about yourself, that would be great.
Yeah, sure. Thanks, John. It's pleasure to be on the podcast, right? And I really appreciate the collaboration effort that we have between KCAS and Sciex. My team, especially Shani, is probably working pretty close with Down for a long time, and also Ian from our tech product team. So first of all, thank you so much, and it was really a fruitful collaboration. Talking about me, yeah. So my association with Sciex actually started like around 12, 13 years back. Started as a core scientist working in a demonstration lab. And my current role at this moment is basically manager, senior manager, scientific marketing, as well as Echo Center of Excellence. So I'll come back, come to that later on, like what is a Echo Center of Excellence, right?
Sure.
So, yeah, my current role, I'm majorly responsible for driving collaborations across the globe. So, as I mentioned, it's very actually pleasure to have KCAS on board, and we have done some amazing projects together. But yeah, I'm majorly responsible for running this. So we work with pharma industry across the globe. I'm responsible for pharma industry, but yeah, we work across all the industries. I partner with them for conferences. We partner with them for publications. We partner for them with them to generate joint marketing content. Basically, the whole purpose is to help the scientific community, right? So that and keep them updated with the new technology, and also try to solve all the challenges across the industry. Before getting into this role, I worked as a demo scientist for almost 8 and 9 years. I was also basically a global lead for pharma applications for Sciex. So all the new feature requests or all the new challenges, right? I used to work very closely with the scientific community across the globe. And now coming to the Echo Center of Excellence. So almost like 4 years back, we introduced a new technology, which was actually a very high throughput solution for drug discovery assays. I was fortunate enough to use one of the first instrument we ever produced. So how it works is basically it's an acoustic ejection, so it's basically replacing the LC for your high throughput applications, and we have a back end mass analyzer. So we started with 6500. What it does is basically just use sound energy to generate droplets, and then transferring that sample to the mass spec for analysis. And talking about speed, that technology is basically only focused on high throughput applications. So if you are regular discovery assays where we talk about net stabs or PK studies where your regular high throughput LC runs around 1 minute, this technology can give you a data at 1 second per sample.
So I put my hand up there. Can you what's the front end to it again? Is it a flow injection based
Yeah,
approach or
it's a
oh,
very high throughput flow
okay,
injection, but there are like no mechanical parts. So what we are doing is
sure, yeah,
basically we are generating the droplets using sound energy,
okay,
and those droplets are captured by open port interface. So basically just catching the droplet in the air and then
mm-hmm.
transferring it through the carrier solvent to the mass spec. So the
Okay,
major advantage, because we know like in discovery, usually the concentrations are pretty high, especially
sure,
when we talk about in vitro incubations. And with conventional LCs, the major challenge that we always see is a huge carryovers, right?
sure.
Because in the discovery setup, you have variety of molecules. Some are sticky, they stick to the LC, and you see huge carryovers. So we basically eliminated that completely. So in this case, there are no carryovers, and your sample throughput is like 1 second per sample.
Okay, okay.
So are those mostly used? I mean, I would love to take advantage of that, but I think what I find, in bioanalysis is unfortunately today we're a little more dirty on our samples and still unfortunately need some of that chromatographic separation.
Yeah.
So are most of the people or your clients using those in more clean samples or, like I said, in vitro screening or something like where are you using that mostly from where are you selling that area?
So at this moment, it's mostly in vitro screens, right? So I know like we see a lot of matrix separations, right? That
Hmm.
actually needs us to have some kind of column hooked up to the mass spec, right? So that we don't see those matrix separations. But when you're talking about echo, actually we the dilution factors are super high, right?
Okay,
So we are injecting in nanoliters. So when you inject in nanoliters in your conventional flow rate of 400 to 500 microliters a minute of your mobile phase, is your dilution factors are way higher than your conventional LC. For example, if you're running um, flow injections on LCMS, you still have to inject in microliters, right? So that the dilution factor is almost 1,000 times higher with echo. So that actually take care of your matrix separations. So you see minimal matrix separations, but again, the trade-off is between sensitivity and throughput, right? So
so
in some of the assays, where
Yes.
sensitivity is not your primary concern, yeah, echo is way to go. That's why I
Yeah,
told you,
yeah.
like all discovery assays, right?
Yeah, so I assume like you're talking like meg per mil concentrations that you're diluting down within the stream.
Yeah,
Yeah,
so that's within the stream. So
you're
if
sure.
if your concentrations are pretty much like double digit nanomolar plus, then yeah, echo is great. But if you're dealing with like picomole concentrations or picograms per ml, then yeah, definitely LCM is the way to go.
Interesting.
Yeah.
So obviously, yeah, I mean, we're as a bioanalytical group, these are the sort of technologies that are nice to hear about, and we get interested, but we, it's a long way being practically applicable to our kind of business.
I mean, it's almost, sorry, John, it's
No,
almost
that's
like
fine. Let's
it's
go ahead.
frustrating more than I mean, it's interesting, but then it's like, well, I can't take advantage of that. So it ends
Sure.
up becoming frustrating sometimes.
Well,
But
and we've had a lot of technologies over the years that have tried to get into this, the speedy turnaround. I know, Jack Henyon used to talk a lot about, these very rapid cycle times that he would demonstrate in his lab at Cornell. And
yeah,
then, I mean, there was the phase where there was a real push for labs like ours to go for sub 2 minute run times until people became more aware of matrix effects. And then I'd been the company I was with previously. We had the nanomate, where again it was looking at nanospray and trying to do that by flow injection, but it still couldn't match what we needed for true quantitation. So it was like a nice idea that we never really managed to make fly in our kind of field. But yeah, you're right. The quicker we can turn things around would be great if we could, because
yeah,
it's
for sure.
we
Especially
can.
like you said, the carryover is always a big problem.
Yeah.
So you can avoid that. Even that new system, I can't think of it. That's like a nano system that it puts all the samples on a spot, essentially, not like a multi spot, but like a little cartridge online, and so there's no auto sampler per se. That's pretty cool too. So some of those technologies are
Yeah.
yeah.
And another thing is like, so we have actually taken it a step up, right? So if you look at like the when you're getting like an output at such a high speed, you need some software, right, to support that data, right? So if you
Mhm.
know, like last ASMS, we actually announced our collaboration with a company called Mass Analytica. So we are actually developing a software that will give you all the endpoint calculations on the go, especially for your discovery assays, right? So if you have an echo for sample analysis, and it will be paired up with the software, we're calling it like AI quantitation. So what it's going to do is going to process your data, and by the time on the go basically, and give you all your endpoint calculations like D-halves, clearances, and all that stuff. Uh, right at the same time. So this is just a glimpse. Like that software has so many other features that basically also designed to support the new interest that we are seeing in the industry at this moment, where people are moving towards high resolution mass spec quantitation, right?
Mhm.
Using TOF systems for the complex assays, especially, right? Um, so that software is also made basically that collaboration with Mass Analytica is also addressing the challenges that we see towards quantitation using high resolution.
Yeah, I mean,
So
I think that applies across the board, right? There's a big interest right now in AI, right? And maybe we don't directly talk about all that today on the podcast, but it does bring up a question, right? Because two pieces are critical to a bioanalytical lab, right? One is getting top of the line instruments that are super sensitive, whether that's triple quads or high res, which we can discuss. But
yeah,
the second part, where we spend a lot of time, is on the data analysis. And obviously, we spend more time on high res data analysis than triple quad. But if
yeah,
you have hundreds of samples and multi analytes, you're going to spend a lot of time picking your peaks and that type of stuff. And so it's definitely, the whole AI field is a hot topic, very growing area where it's nice to hear, instrument companies working with them, because we don't, as a CRO or a pharma industry, don't want to have to reinvent the wheel all by ourselves all the time. So that'd be nice to take advantage of.
Now, this is like, yeah, high res is definitely adds advantage, right, for your complex bioassays. I still remember like the first time, like especially with the new instrument that we launched was Xenotop 7600 system, right, with the trap, and we tried to match to the sensitivity levels of triple quads where and also the stability, right, because that was always of one hesitation that I have seen with all the CROs that, oh, can it really do as good as my triple quad, or can it really do as good as Sciex triple quad, especially when they're known for robustness, right? So we always heard that, and I was super happy. I remember like first time I saw a beautiful peak coming out of high res mass spec at 1 picogram per ml, which actually I never thought about that when I started career with mass spec industry. I was like, yeah, no, I don't think we can get there, but yeah, it's moving pretty fast.
Well, I
Go
think
ahead.
that actually is it's a really good point because I've made this point several times on this podcast how quickly the technology has evolved with what we've been doing. I mean, I've been in the field 20 odd years, and the change in the technology in that time has been phenomenal. In fact, going back to my PhD, that you wouldn't recognise the mass specs then compared to what I work on now. But one of the things we wanted to talk about was the Sciex history and their focus, and then the partnership with pharma and biotech. So maybe this is a good way to talk about this because I mean I'll confess, the last Sciex instrument I actively used was the API 3, and I
Yeah.
think if I remember correctly
Don't date yourself
that
there,
came
John.
from the Taga, which was more of a gas measurement instrument, and then obviously it became the gold standard for LCMS at the time, and things went from there. But Rahul, do you want to talk a bit about Sciex history and
Yeah.
just the
So
company philosophy? Because I think that's it's helpful to have. I mean, I hear where you're coming from with the echo stuff, and obviously you're meeting the need in a certain part of the mass spec space, but to sort of talk about how Sciex has been integral to bioanalytical, it would actually be really valuable from your perspective.
yeah, so talking about history, Sciex history with mass specs, right? Especially triple quads. I think just few years back we celebrated our 50 years. So,
Mm-hmm.
so we have been working aggressively, and I'm pretty sure like pharma was definitely one of our core market, and still it is, right? That's where basically we are making people's life better and making a change to overall health industry, right? So yeah, we are still focusing on the same way, and I would say the one of biggest strength of Sciex from the day one was giving people a mass pack that they can run day, night, and out to basically improve their turnaround times and come up with the solutions to help the health industry at much faster pace, right? So we are still on the same path, challenging ourselves every year, coming up with more robust, more sensitive products so that basically it doesn't impact or there are low down times, right? So it basically helps in the health industry to move towards the solution at the much faster pace. Like recently, I went to one of the big CRO lab in India, and I was so surprised. He took me and he told me like, "Oh, there's a surprise for you," and I was like, "What?" He was like, "Come, I'll show you." And they still had like API 2000s running.
Wow.
So
Yeah.
so it's so happy, and with all the collaboration that we had with KCAS, Down has been amazing with the new technology, the 7500 plus, right? And I tested that instrument in my lab. My team played with it, and I'm sure Down is running that day, night, and out. And we are still maintaining, like we have improved on the sensitivity thousands times from those machines, and it's always give you so much satisfaction to see the scientific community still connects themselves to the same technology with the same trust, right?
Yeah, and
So
I'm gonna jump in for a minute. And I know Don wants to say something. One of the
yeah,
things that one of the reasons from a customer perspective is that obviously we're all trying to stay on top of the current technology, but once you get an assay validated on a platform and it's running thousands of samples, it sits comfortably with that platform, and you may not need the additional sensitivity, which is why we end up with a lot of legacy instrumentation while still looking for the next platform, because we know that the way the different sort of moieties that you're seeing come out of the pharma company, you're likely to have more challenges in terms of sensitivity. So you always want to be going for those better or those higher end instruments. But I'm going to give Dawn a chance to talk
Yeah,
to.
I just was just gonna. It made me, reminisce right about history, like you said, API 3. So when I started back in the '90s, I was on an API 3 as well, and it was on a Mac platform. And when I was writing
Yep.
my dissertation, Macs and IBMs didn't really talk nice to each other, so you had to use converter programs, and it was very, very painful just to see the evolution of a company or an industry is pretty amazing, right? And you had to like cryocycle it every night, and then you're down for 15 hours or whatever it was, right? So it's very nostalgic when you think about it. Now they just run 24/7 all the time and are super sensitive, and software's better, and it's very, very different. It's reminiscent of maybe my parents or people saying, remember that computer back in the '70s that had took up the whole room and it was cards, and it just is kind of funny like that, I guess.
yeah, that's actually, I would say, down, you got it at the right point. This is my best picture I ever got clicked in Sciex was right by API 3 in Concord.
Yeah, it's kind of funny. Yeah.
Yeah, it was a great instrument, and certainly for years there was like everyone was scrambling to catch up with it. But in terms of sensitivity, but of course, like you say, Don, you had that because of the cryo pumping, you had those downtimes where you had to let it recycle to then be able to pump again and actually run more samples. Yeah,
Yeah.
it's a fun instrument to work with. We used to try and push it as much as we could. So we were at ABS where I was. We had three API 3s, and we knew roughly the time that each would go before it got to the point where it would automatically go into recycle. And so we tried to juggle as many runs as we could
Yeah.
before it hit that point. And obviously, like say, once it went into recycle mode, then you were done. But yeah, because we had one assay in particular. It was an approved drug that had a number of active. So it was a pro-drug, number of active metabolites, and it was so we had multiple assays that were running. One of which was a 15-minute assay. So there was a lot of stuff we're trying to do, and it was all on API 3s. And so, like I say, you couldn't afford to be down every night. You needed to be running overnight. So that's why we were running, like, I think our best one was about 45 hours. Can't remember the other two, but it
That's
was this
funny.
sort of we really pushed the instruments to the limits in terms of how we used them. But like I say, it really was the gold standard for a long time. And so then for as a customer perspective, the 3000 for Sciex was the next big thing, and then the real game changer was the 4000 onwards. And I'll back off and let other people talk.
Yeah, even it went to orthogonal, right? A better spray
Yes,
didn't get
exactly.
as
That
dirty.
change of ion source made such
Yep.
a difference to our ability to run samples and not lose the instrument for, because it needed cleaning and all that kind of thing. So it was it was the switch in this sprayer really transformed our lives.
Well, and maybe again we can lead into some of the newer stuff now, right? I mean, now we're on the 7500, the 7500 pluses, right? And we have a number of those, and we use them 24/7, right? The advantage of the plus so far in our experience, right, is it's supposed to be a little bit more robust, a little bit more rugged, easier to clean, lasts a little longer before it gets dirty, and for us, that's really critical as a CRO, because any downtime is time and money and client unsatisfaction, right? You don't want any of that. So we want to always keep things running to the best of their ability. And instruments, a lot of times, your playing with it, tweaking it, getting it to be its perfect amount, perfect sensitivity, and then you run it for a couple of days or a week or a month or whatever, and if it starts to taper off, that's a problem, right? So if we can have things last longer, more consistent for a longer period of time, that always helps us quite a bit. So we really appreciate that effort
Yeah.
and collaboration.
So, yeah, just to add to your point down, right? So I remember for like so many years, whenever Sciex used to go to ASMS, right? People always expected us from us like, what is the next level of sensitivity, right? So when we came up with the 75, though we did pretty great, and it's very important, right, to listen to the customers, and especially when the sensitivity has gone up like hundreds times compared to the old generation systems, right? The robustness, resilience, right, is pretty important, right? So that was the whole purpose coming up with the 75 plus, because we took the feedback and it's like, okay, sensitivity wise, pretty good, damn good. 75 is doing great, but can you make it more robust, right? That was a challenge. So our R&D team took it, and probably the last ASMS was the first time that Sciex actually came up with the product that was actually specially designed for enhanced resilience and still keeping the same sensitivity level of 7500, which was actually industry leading and still industry leading, right? So, yeah, it's
I'm
just,
still waiting for that 8500. I'm always pushing sensitivity, so
Yes.
I will keep knocking. I'm one of those, give me more sensitive, give me more sensitive. So I'll always knock on that door. But
So
well,
Rahul, for someone who's been out of the lab for quite a while, what kind of changes were made between 75 and 7500 plus? Was that more front end, like increased the different gas
yeah,
optics,
so
or how was it?
we,
How?
yeah, so what we did is basically we introduced the mask guard technology, right?
Okay.
So what we realized, like over the time, that the major thing that which was responsible for your quad contamination, right, like Q1, was actually the larger molecular weight ions that are
Mm-hmm.
coming from the sample, right? The small molecule contaminants you can wash it off with SPE, right?
Okay.
But still, there's tons of proteins and peptides that go into the mass spec along with your ion of interest, right? So those were actually more responsible for contamination of your quads. So that's why like we came up with the mask guard technology. So what it does is basically it functions as a cutoff, right? So
Okay.
whatever mass you are looking at in your target analyte, it will do a mass cut of 100 Daltons above it. So basically, we are not letting any large molecule ions going into your mass spec, and that actually enhanced the mass spec's life like way longer. Like you don't need a routine maintenance or cleaning on the 7500 plus. Like frankly speaking, I did like a pretty strong accelerated testing of this instrument, and I actually had to give up internally when we were testing it. Like, okay, this is not going bad. We have to stop it, right? And we actually tested it like twice, almost twice more compared to the 75. But maybe Don can speak much
Yeah,
more.
I was going to say it'd be interesting to get Don's perspective on that
Yeah.
as
yeah, so
the expert.
we have about, I would say, 10 of the 7500s, and we have 3 of the pluses formally, and then we've had this collaboration with Sciex where we've been evaluating one before it was formally released. And kind of to that point, we've, injected lots of different samples. So we just treated it like a regular instrument, let it see everything that it can see from, relatively clean to not so clean to dirty, whatever. And I would say it does a pretty good job. It may be in that 2x range where before we have to, clean it,
Sure.
pull the rail, that kind of thing. It's hard to say though, because you're not exactly running. In the beginning when we did the collaboration, we literally did exact sample splits, right? We would take a
Sure.
sample set, run the exact same samples, and inject it on both systems, so we had an apples to apples comparison. And that's probably where we saw that 2-ish, X robustness, ruggedness kind of thing. Now we're running it not quite, double dipping, because we don't, can't repeat everything all the time just from a time perspective. So it's a little bit not quite an apples to apples, but it definitely has a longer performance for sure. The interesting thing is I do find, different instruments sometimes just whether it's the nature of the instrument, call it a hot box or whatever. We do have some that just seem to do better, but maybe it's, the assays we're putting on it may just last a little longer. They're a little bit cleaner, something like that. So it's, can't directly pinpoint it, but definitely is an improvement there. I guess one of the things I wanted to ask, so, we've had a lot of recent conversations. Back in the day, we did a lot of nano, right? We had easy or spray, nano spray 3 or the different sources, and Captiva made a source for Sciex and different sources that we could run nano spray on. And normally we would see a 10x, 20x gain, let's say over high flow. And then, you guys went to the OptiFlow source, and then there was a big push, maybe 5, 6, maybe even 10 years ago on microflow, and people moved to microflow because it was a little bit more robust, right? Same for the plus, right? You want something ultimately in a production lab or in a large lab that's robust and not so finicky. So we kind of went to micro. And maybe with like the 7500s as well, you have better heaters, you ionize better, right? One of the benefits of nano was always, maybe in the old days, the sources didn't ionize high flow completely, so nano was more efficiently ionized.
Yeah.
So maybe, Rahul, you can speak a little bit to that, because I feel like, the 7500 doesn't directly support nano as much anymore, and maybe it's a dying art or maybe it's not. But, as somebody who's always pushing sensitivity, like I was joking about the 8500, but however I can gain sensitivity, whether it's sample prep, whether it's chromatography, whether it's instrument performance, maybe you can speak a little bit to Scix's thoughts in that area.
I think also my additional question to that is what other needs do you hear from other customers where you might like to go in the future? We've heard about obviously the instruments being more robust in terms of lasting longer between cleans, etc. That's obviously something we all want, and obviously the nano micro thing is another push. Where else do you hear that kind of feedback? I would like to hear the answer to Don's question first.
Yeah, no, absolutely. You touched the right thing, right? Evolving needs of bioanalysis, right? So, yeah. So what we do is basically first to answer Down's question, right? So basically 75 plus, right? It was clearly built keeping our CRO consumers in mind, right? Where you need more resilience and basically keeping still the high standards of sensitivity. You're right. It's at this moment, it's not directly compatible with Nano, but we did some testing initially. The source flow dynamics on the 75 plus is really good. That we actually we are pretty close. We almost bridged that gap from going from like nano to micro. Like you can easily see up to 5x gains with the very well optimized microflow method running at 2 to 3 microliters. Nano compatibilities, yeah, it is there with our accurate mass systems to basically support proteomics customers, right? But you're right, like since micro is more robust and this was triple quad, and especially keeping in CROs in mind, we felt like maybe the CROs are not much inclined towards running nano, but yeah, if you feel you need it, you're heard, and maybe now we will have you on the collaborate with us on the nano stuff as well.
Keep
Yeah,
on
yeah,
keep on his case, Don.
well, yeah, maybe to that point, right? I think EvoSep was what I was thinking about earlier, right? The non-auto sampler carryover kind of thing. And I know I do think Sciex works with them some, and again,
Yeah.
that's kind of a nano thing, so could be something to look at. But I agree with you. Generally, we're at the mercy of our clients, just like
Mhm.
you're at the mercy of your clients, right? And when they need what they need, we try to help them, solve their problems where they can. In discovery, it's a little bit more forgivable to do nano or micro, but I agree. Like I always try to push back too, because it's not as robust when we go to a production high throughput. We want something day in and day out that's going to be robust and rugged and not have to be artful and switching sources back and forth all the time, because again, it equates to downtime, right? So yeah,
Yeah.
for sure. If there's an easy way to do it, I would absolutely be up for the collaboration.
And on John's question, right? What we hear more from people like now, I think what we are hearing with definitely with the drug modalities getting more complex, right? We are talking with products. We are talking about all
Mhm.
those drug conjugates, right? The first thing that I always hear, like from the customers, like, "Oh, when are we getting accurate mass which can do as sensitive 7500, right?" So I was like, "Sure, we are working on it." That's we always keep on challenging ourselves, right? With Zeno top 7600, we were pretty close. We tried to bridge that gap, and definitely, Sciex one of the top priorities to listen to customers and keep working and moving. But yeah, we are seeing a huge trend where it's not like triple quads, but people are adopting, and especially CROs are a little bit opening up towards the high res mass spec for quant, as well as they are pretty good for doing your qualitative med ID studies, right?
Yeah.
Especially, for example, with 7600, we came up with electron activated dissociation, where basically you can control the electron energy, and that can you can run just any kind of molecule with it, right? So and it's really helpful for med id, so people feel like, okay, is there a box that can give me like high level of sensitivity and as well as I can use it for my discovery applications? So yeah, it's continuous process. We
Yeah,
keep
I'm definitely in that shop too. Like, so we're, have recently invested in high res and are supporting, several clients with that as well. And, a couple of things come up there. One, I 100% I'm still, I'm maybe on the fence, as a convert of doing triples for so long. And I've done high res obviously since the late 90s as well, but triples are just such the workhorses, right?
Yep.
yeah
And so I'm still a little bit like in that what you just said, right? I think triples inherently are more sensitive, but obviously when there's an interferent or some specific area, then high res can be more sensitive. But I would say even we've doing some recent oligo analysis, and high res there is, in full scan, let's say, is really full scan is good enough because of the resolution and the specificity that we are getting even better sensitivity than straight off the triples. But I definitely, if you could find a really good high res that in targeted quant always gave us, PRM or whatever we want to call it these days, I would be in line for that as well. But I guess the other question that brings up, right, is data file size, right? We're running up against that right now because the file size of I could probably put 6 instruments for 5 years on a flash drive that I carry in my pocket, just because
Don't
it's
tell our IT that.
yeah. No, I'm just saying not from a
I know, I
security,
know.
but from a space triple quad space.
Yes,
It's
yes.
very very small file sizes, and the file size on full scan profile high res, are hundreds of
Mhm.
megabytes just for 5-10 minutes of data. So it's becoming a whole nother work stream of cost and backup and, to your point, John, security, right? How do you have redundancy and backup and make sure, if something were to happen, you have another backup, and it's definitely up on our radar right now.
Yeah, no, absolutely. This is, I hear that all the time that the data file size on high res mass specs is way bigger, right? And especially when you're in setup, for example, you said you have like 137500 or 7500 plus. So it's tremendous amount of data that you're generating every day, right? So I think that's the reason, right? The triple quads are basically the favorite instruments for all the CRO setups, right? Smaller data file size
Mhm.
and still getting what you need, right? Your endpoints that you need, right? So yeah, hopefully with the technology advancement, maybe we'll come to a point where we will be able to make shorter or smaller data files coming out of high-res mass specs.
I wonder if AI could help there at all. I don't know. Maybe it could intelligently suck out the data we needed and make them smaller after the fact or something.
Yeah, maybe it's a good idea. Maybe
Yeah, who
we can
knows?
talk about that offline.
There's your next collaboration in line, I suppose. um, I know that we have to be sensitive to time. Is there anything else that you would like to talk about, Rahul, in terms of what Sciex are doing at the moment or planning for the future, and then we can wrap up and obviously respect your time today.
Yeah, no, we almost touched everything. I really loved the discussion. It was amazing. Yeah, at this moment, as you said in the beginning, right? So the focus is team is on the way to WRIB next week, and yeah, looking forward for the next ASMS, and probably would see down there as well. She was there for us last year. So we'll and then yeah, can we continue working on the needs of the market and try to solve the complex issues that we are seeing in the industry. So thank you for having me today. It was pleasure talking to all of you.
No, you're very, very, very welcome. Hopefully, sometime I'll do ASMS. I'm not sure it will be happening this year, but I'm long overdue getting back to that meeting to see what's going on. But
It's
yes,
definitely
it's been a
changed
real
a little bit,
pleasure.
Don. It's not quite as fun as it used to be,
Yeah,
but
sure.
there's still a lot of good information.
That's all right. I'm so much older, and I'm not as much fun as I used to be either. So I think
I'm
we're
always
still
fun.
a
I
good
don't know.
match
I'm
on
always
that point.
fun.
Yes. In that case, I think in that case, like I say, I want to respect your time, Rahul. I really appreciate you joining us today, and thank you very much. And yeah, it's been great. Thank you. Is there anything else you want to say, Don?
No, yeah, no, I appreciate it, and would love to continue the collaboration anywhere I can, and hopefully it's mutually beneficial. And I'm always a tough customer, but hopefully give good feedback, and look forward to doing that more.
So
Yeah,
Rahul, one thing we got a couple of minutes. We usually do like some hot seat questions just to get a sense of people's personalities outside of science, etc. I'm going to jump over anything, but as we're coming up to the weekend, and as I'm in Poland and thinking about what I'm going to do for dinner tonight, I was just what's your weekend plans? What do you like to do in your spare time outside of Sciex, etc.?
yeah. It's like I have a 6-year-old daughter, so she has just started gaining interest towards sports. So
Okay.
that's my weekend activities these days.
So what sports does she go for?
Oh, I come from India, though, so she is trying to look for something which is very close to cricket. So
Hmm.
softball,
Okay.
that's her
Hmm.
target now.
I'll give you some pointers. I coached softball for 15 years, and my girls just finished. I have 2 years ago, I haven't done it for a year or so, and it's very sad. So enjoy it while it lasts, because eventually it ends, and then you get a little sad.
Well, thank you very much. On that note, I think we should wrap up and see you next time.
Yeah, pleasure.
Very good.
We'll see you next time for sure. Yep.
Thank you. Bye.